In this video, we're going to introduce metabolism, taking a look at anabolic and catabolic processes, and then looking at oxidation and reduction reactions, and then finally the mitochondrion, which is going to lead us into our first look at a metabolic process, which is cellular respiration. Metabolic pathways consist of enzyme catalyzed reactions. So enzymes are causing the reactions to occur, and they occur through several interconnected steps and stages. So for example, if we look at aerobic respiration, which we'll be spending a lot of time with, it involves 4 different stages, and each of them have several steps within them. So metabolic pathways aren't just one simple reaction, they're many reactions made of many steps and stages. Some of these steps form cycles where the end product acts as the reactant, and an example of that would be the Calvin cycle or the Krebs cycle, which are each components of, for Calvin cycle it's part of photosynthesis, and the Krebs cycle is part of cellular respiration. Now, metabolic pathways can either be catabolic or anabolic. So this first reaction here, where we have 2 reactants coming together to form a product, that would be an anabolic pathway. So an anabolic pathway is when you're building up, so you're making a larger molecule from smaller components, whereas a catabolic pathway would be breaking down. So this molecule being broken down into these would be a catabolic pathways. So anabolic and catabolic. So enzymes' job is to reduce the activation energy for a reaction. So they lower the activation energy, making less energy required to start the reaction. So they're speeding up the reaction without getting used up, and they do this by reducing the amount of energy needed to be put in to start the reaction in the first place. So in this graph, we see here, if there wasn't an enzyme, this is how much energy it would take to get the reactant started and then off to products. Now, if there is an enzyme, the activation energy is much reduced, so we can see that in the dotted line. So there's not as much energy expenditure to start the reaction. So that's what enzymes' job is. And if you look here over time, you go from your reactants to your products. So one thing we need to explore is the effect of enzyme inhibitors. So in page 376 in your IB biology textbook, this diagram is found. So if you take a look at the first one, so if there's a competitive inhibitor, the reaction would be able to eventually meet the rate of reaction of the normal enzyme if there's more substrate added. So, at normal substrate rates, it would be inhibited, but if you added more and more substrate, eventually there would be enough substrate to be to overcompensate for the inhibitor, and the reaction rate would catch up. But with a non-competitive inhibitor, because it's binding at an allosteric site and changing the shape of the active site, it doesn't matter how much extra substrate you add, it's never going to get to the rate of the normal enzyme because the active site's shape is changed, the substrate can't bind at all. So you're not going to be able to get to the normal enzyme rate, which is this purple line here. In many reactions, we will observe end product inhibition, and what that means is that in these reactions, the final product, so in this case it's this one, C, will allosterically inhibit the first enzyme. So it'll bind to a site other than the active site at the allosteric site, change the shape of enzyme 1, and then we can't act on the first substrate, and so the reaction can't happen at all. So the pathway stops. A real-life example of this would be the reaction where threonine, which is an amino acid, is converted into isoleucine, which is the end product, and there's a whole bunch of intermediates along the way. And as isoleucine levels rise, so that isoleucine is your end product, it will bind to an allosteric site on enzyme 1, and it'll inhibit it, and then it stops the pathway, so no more threonine can be converted into isoleucine. So more and more scientists and researchers can use databases to collect information and treat a disease or solve a problem. So bioinformatic databases can actually be used to sift through possible treatment options for disease. And the reason why is that most drugs block some form of metabolic pathway to make them effective. So if you can find a drug that's going to inhibit one part of a metabolic pathway that you're certain that the disease takes, then you can use that drug then to treat that disease. So malaria would be an example of where that has been successfully done. So redox reactions are also known as oxidation reduction reactions, and they involve the transfer of electrons from one substance to another. So there's a couple of ways to help you remember that. Reduction reactions is the gain of electrons during a reaction, so GR. And then oxidation is the loss of electrons, so that we say is LEO. So sometimes the way you remember it is the loss of electrons is oxidation, and the gain of electrons is reduction. So Leo, the lion, goes grr, helps you remember the two statements. Now this table handily compares both oxidation and reduction to help you remember and be able to track whether something's been oxidized or reduced. So what you have to do is look at the molecule on the reactant side and then look at the closely related molecule on the product side, and then you can take a look and see what's happened. So if from the reactant side to the product side the substance gained oxygen atoms, then it's been oxidized. Another way to know this is electrons have been lost from the substance, and at the same time when the electrons are lost, so are hydrogen atoms or hydrogen ions. And when all of those things happen, the substance loses energy from this loss of electrons and hydrogen. So oxidation involves a loss of electrons, hydrogen, and a loss of energy. Reduction reactions, you remove oxygen atoms from a substance, or the way we will trace things will be gaining electrons and gaining hydrogen atoms from a substance. So in this case, when we can trace the electrons and hydrogen, we know that the substance has gained energy from the gain of electrons and hydrogen. So that's the easiest way to identify if something's been oxidized or reduced. So right now we're going to take a look at cellular respiration and explore what items are oxidized and which ones are reduced. So if we look at this molecule right here, which is glucose, we have C6H12O6. The carbon-containing compound on the product side is CO2. So we can see that glucose loses hydrogens. So in this case, this molecule has been oxidized to become carbon dioxide. The other molecule is oxygen. Oxygen gained hydrogens to become water. So this molecule has been reduced. So remember, if something loses hydrogen, it's also losing electrons and losing energy. So this went from a high energy molecule to a low energy molecule. And then this one went from a lower energy molecule to a higher energy molecule because it gained hydrogens and gained electrons. So phosphorylation is when you add a phosphate to a molecule. And many biological molecules have binding sites for phosphate groups. And when a molecule gains a phosphate, it makes it more unstable. It makes it more likely to react. And sometimes we want molecules to react, so it's advantageous to gain a phosphate. So a very important molecule that we'll be talking a lot about in this unit is ATP. And ATP is short for adenosine triphosphate. So it has adenosine attached to ribose, and then attached to it are 3 phosphate molecules or phosphate groups. And this molecule combined is known as the chemical currency of the cell. So in our economy, we use money to purchase things. So to buy or sell things, we use money as our currency. In the cell, anything that needs to be bought, so anything that requires energy, anything that has to happen, you have to spend energy. And you have to spend the energy in the form of ATP. So ATP is a very important molecule that we'll be spending tons of time talking about. So ATP provides energy to molecules when it releases its third phosphate to the other molecule. So that phosphorylation. And when it loses the third phosphate, it goes from being called ATP to ADP, adenosine diphosphate. And that phosphate will be sent to another molecule to drive a reaction. So the last thing we're going to talk about in this video is the mitochondrion, because starting after today, we'll be spending time looking at cellular respiration. And one of the organelles responsible for cellular respiration is the mitochondrion. That is for aerobic cellular respiration. We will take a look at anaerobic respiration, which does not require the mitochondrion at all, but aerobic respiration does. So the mitochondrion, you may have learned in grade 10 science, is the powerhouse of the cell. So that's not a lie. It is the organelle that creates a ton of ATP, so it creates a ton of energy for the cell to do all the things the cell needs to do. And now we need to know the internal structures of the mitochondrion. So if we take a look here, this outer part is the outer membrane. And it actually has two membranes. So now we have the inner membrane here, and it's more folded. So we have a smooth outer membrane and a folded inner membrane. And then in between the inner membrane and the outer membrane, so in this space here, we call that the intermembrane space. We'll spend more time talking about that a little later. The fluid part, the yellow part here, is the fluid-filled space of the mitochondrion, and that is called the matrix. And many of the reactions of cellular respiration happen in that area. Now, we talked about the inner membrane being highly folded right here. These folds have a special name, and those folds are called cristae. So the folds are called cristae on the inner membrane. And that is the mitochondrion. So this is an electron micrograph image of a mitochondrion. So right here is the mitochondrion. And you can see this is the outer membrane here along the perimeter of the organelle. And then you can see all these foldings inwardly. So these are the cristae of the inner membrane all along here. And then in this space here, that would be the matrix, or the fluids-filled space. So that would be your mitochondrion if you were to recognize it as an electron micrograph. The really distinctive feature would be these long stripy lines inward that don't go all the way across, so you know that there's folds partially. So this is how you could identify an electron micrograph of a mitochondrion.